All right. So I'm really faded out in this one. Maybe it'll be better for y'all. Um, all right. So this is all about cancer, not my favorite topic. I wish it did not exist, but unfortunately it does. And so we're going to go through our ICANN statements, make sure you have a pretty clear understanding about cancer and the things that cause it, and some differences and things like that. So first of all, I can define cancer. Cancer is uncontrolled cell division. It is basically mitosis that's gone haywire. And that's why it's so frustrating, because it's your own cells. So that's why your immune system doesn't always recognize them, because they're like, hey, these cells belong here. No, they don't. We don't want them there. But anyway, that's what it is. It says the next ICANN statement: I can describe the processes that lead to transformation of a healthy cell into a cancerous cell. And I'm going to get into a little bit more of this later when we talk about proto-oncogenes and tumor suppressor genes. But just at this point, what I want to say is it is due to genetic mutations. And so while it could be one mutation that leads to cancer, that does happen sometimes, depending on where that mutation is. A lot of times it's more than that. And so the average amount of mutations it takes for a healthy cell to transform, and that's what we call it when it converts to a cancer cell, we say it is transformed, is 5 to 7. So 5 to 7 genetic mutations. And those mutations can be in all kinds of different places, but basically they're going to be in the regulatory system that controls cell division. And so when cell division happens faster than it should, that's because something has gone wrong. Something that should be controlling it is not. And so that is what's going on. And so one of the things that healthy cells, our next one says, I can define density-dependent inhibition. One of the things that our healthy cells does is, there we go, is our healthy cells do is when they are touching another cell, they get a signal that says, hey, you know, there's not much room. We're kind of crowded in here. And they know not to divide because there's not room. And if there's not room, that means there's not enough resources. There's not enough oxygen and food and all that good stuff. And so they stop dividing when they're touching each other, because they know, hey, we've filled up all of our space. We're good. We don't need to do anything. And so that's what you see here. So they've covered the whole mat of the bottom of the plate. And so when you take the cells out, they will fill that back up and they'll stop growing again. That's density-dependent inhibition. They inhibit their growth based on the population density. So when it's really dense, they stop growing. Cancer cells don't do that. They lose their density-dependent inhibition, where they will just continue. You can see here in this diagram, they just start growing on top of each other, and that's what a tumor is. The other thing that normal healthy cells have is anchorage dependence, and that's your next ICANN statement. I can define anchorage dependence. Anchorage dependence simply means that in order to divide, the cell has to be anchored to something, either another cell or the extracellular matrix. And so in this picture, you see that these cells are anchored to the bottom of the plate, which they treated to make them think is the extracellular matrix. And so they did divide and fill up that space. If they had been free floating, just in like liquid agar or liquid nutrients, they would not have divided, because normal cells don't divide in that situation. Cancer cells can. And so they lose their anchorage dependence. And those are due to mutations of the genes that control those, that cause that density-dependent inhibition and that anchorage dependence. Those genes have been mutated, or those signaling pathways there's a mutation in one of the molecules in that pathway, because both of those are signaling pathways. So that's what those two things are. I can compare benign and malignant tumors, and I can contrast them. So a tumor is just a growth of a lump of cells. And so when that lump of cells sticks together and stays in that site, we say that it is benign. And so all we have to do to get rid of it and cure that tumor is scoop it out and take it out of the body, and it's cured. Now, this isn't always possible. I have a friend from high school whose mother had a tumor in her brain. It was benign, so it wasn't going anywhere, but just because of the location, they were not able to go in and get it, and it was dangerous because it was pressing on sensitive brain tissue. So benign tumors aren't always healthy, good, but they're not going to spread. And so that's the benefit of those. But depending on where they are, they can be a problem. Technically, depending on some people's definition, some people call a mole a benign tumor, because it is a growth and overgrowth of some cells in an area. It's not spreading. It's not doing anything. It's not, giving you any problems. It's not technically, we don't consider lay people like us don't consider that cancer. I don't know about doctors. It probably depends on the doctor. But for the most part, when we hear benign, we think, oh, good, they don't have cancer. So that's kind of one way to think of it. Malignant tumors, malignant means bad, comes from the root word that means bad. These are tumors that are growing. They are, not that benign tumors aren't, but malignant tumors are actively growing, but also pieces of them can break off and go into the bloodstream and move to other parts of the body, or just send out tentacles. I had a cat that had cancer, and his cancer had tentacles. And so even going in and taking out the tumor only solved part of the problem, because those tentacles were beyond the where they took out the cancer, the tumor. And so malignant tumors are going to spread, and they are going to start impairing functions of organs. And so that's what we call cancer. So generally speaking, we think of benign as good, not cancer, and malignant as bad. It is cancer, but like I said, benign is not always good, depending on where it is. All right, I can define metastasis. Metastasis is just that spread of cancer. It's when that cell breaks off, travels to another part of the body, and starts a new tumor. That's all metastasis is, and the verb form of that is metastasize. So you might hear that as well. I can discuss the role of proto-oncogenes. I did want to go back. Make sure you look through these PowerPoints or Nearpods, because it does talk about some different ways that cancer cells get past normal processes. So make sure you look at that. But anyway, going into proto-oncogenes and tumor suppressor genes. Okay, a proto-oncogene is a good gene. You want those. Proto-oncogenes are genes that stimulate normal cell division. So when a proto-oncogene is healthy, it's not been mutated, you're going to have normal cell division. It's kind of like the gas pedal, though, on cell division. So it pushes the gas pedal to tell the cell, hey, you can divide in a normal, healthy way. And then you have tumor suppressor genes that stop cell division, and that's how they suppress tumors. It's not like they're actively looking for tumors and they kill them. It's that because they're not allowing cell division to occur, they're not allowing tumors to develop, okay? So they're more on the pre-tumor side of things. So they're stopping cell division. So they're kind of like the brake pedal, okay? When you have mutations in these, it's like taking your foot off of the brake and putting your foot on the gas. And so that's when we get to see problems. So I can discuss the role of proto-oncogenes in the formation of cancer. A proto-oncogene, when it mutates to become no longer controlling cell division in a healthy way, it becomes what we call an oncogene. Onco is in cancer. It's a prefix that means cancer. And so the proto means like before the oncogene, and so when we drop the proto, when it converts and becomes a cancer-causing gene. And what it is it's us putting our foot on the gas pedal and saying divide, 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 divide. We don't want that. But that's what's happening when those are mutated. The role of tumor suppressor genes, like I said, they're the brake. And so as long as they're healthy, they're good. But when you take your foot off of the brake, your car is going to roll forward, right? Unless you're in park. That's a whole other story. But with tumor suppressor genes, what you've done is you've taken your foot off the brake. And so if you have one foot on the gas and you take that foot off the brake, it's going to go forward. And so that's when cancer happens. There is a condition. It's really an interesting condition if you want to do a little research, called Lee-Fraumeni. It's L-I. It's two doctors' names, Dr. Lee, L-I, and Dr. Fraumeni, which I think is F-R-E-U-M-E-N-I. I might be a little off on that spelling, but if you Google that, you'll get close enough and find what I'm talking about. And it is a mutation in a tumor suppressor gene, and these people have high rates of cancer, and so they're always being monitored and to catch those cancers early. So it's a unfortunate condition, but it's interesting that could happen. So it's a genetic condition, a dominant genetic condition. All right, and then I can describe treatments for cancer. So in here, and in the Nearpod, I actually had some extra ones, some more advanced. Don't think they're in this PowerPoint, but I'll add them when I'm done with this video. So treatments for cancer, surgery. So if it's localized, or even if it's if they feel like they can get most of it, they'll usually go in and take what they can. They'll get out what they can. If I have another friend who has was just diagnosed with kidney cancer, I don't know, a month or so ago. It was localized. It had not spread. They went in and took out her kidney, and she is in remission. So those kinds of things can happen too if the cancer is caught early enough, and or if it's a slow-growing cancer. Radiation basically burns the cancer cells with radioactive. Not radioactive. Well, it depends. Sometimes it's radioactive, but anyway, it's radiation, just like you think of with like the sun's rays are a type of radiation. But it damages the cancer cells' DNA so that they basically go into apoptosis because they're so damaged. They're like, whoa, something's wrong with us and we can't fix it. It does damage normal cells around it, but the good thing about radiation is you can really pinpoint that. You can radiate very small parts of the body very easily. Chemotherapy is not like that. Chemotherapy goes into the entire body and it's going to affect every cell in the body. But it is drugs that are going to be toxic to anything that divides rapidly. So cancer cells are dividing more rapidly than normal cells. And so they are targeted with these chemotherapeutic agents or these chemotherapeutic drugs. Different drugs work for different cancers. So there are tons of different types of chemotherapy, and they all work for different types of cancers. And there's different combinations. There's cocktails of chemotherapy where you might take these 2 or these 4 or whatever. It just really depends on the type of cancer, your genetics, all kinds of stuff. And so some of the cool things that we're doing, science, all of us being scientists, not me personally, but this being done right now is things like immunotherapy, where they're taking the immune system out and they're really priming it to recognize that cancer, and then they put those cells back in the body and it goes after that cancer like crazy. Hormone therapy. So if you have a particular cancer that is affected by hormones, they can block or remove those hormones so that it kind of starves the cancer because those hormones stimulate the cancer. There's cryoablation where they go in and freeze the tumors. There is targeted drug therapy where, again, and the whole pharmacogenetics, which is pharmacy, drugs, plus genetics. And so they're looking at cancers and genes and how does it all work together and how can we tailor these treatments, not just to the type of cancer, but to the genetics of the person. And so that's a really exciting kind of forefront. And then there's all kinds of clinical trials. So there's drugs and treatments of different kinds that are being tried that people can go and have those clinical trials done if others aren't working or if that's just the best option at the time. So those are different treatments. Now, what can I do? What can you do? What can we all do to prevent cancer? Well, sometimes you can't. I mean, sometimes it happens. And so don't beat yourself up if it happens. But the big thing with cancer is trying to catch it early. Some things you can do that will decrease your risk. Don't smoke. So anything, and that's legal or illegal. So obviously I don't want you doing anything illegal anyway, but breathing in dirty air is bad for your lungs. Your lung tissue is very delicate. And so anytime you breathe in air that is not clean, you see an increase in lung damage and lung tissue disorders like cancer. Use sun protection. Sunscreen, broad spectrum, so it covers UVA and UVB, 50 or higher if you can. I think that's where they're saying now 45 and 50, SPF 45, SPF 50 is where they want you. Exercise and keep your weight at an ideal level. That just helps you stay healthy overall. It keeps your immune system healthier. Eat fruits and veggies. They have lots and lots of good nutrients, lots of coenzymes and vitamins that help your body work better. And the better your body works, the better your immune system works. And also the better things like the proofreading. When you copy your DNA, that's proofread. And so if those enzymes are proofreading that DNA better, they're more likely to catch mutations. Use screenings. There are so many things we can do to try to catch things earlier. Breast exams. Females should be doing these, and also males, because males can get breast cancer as well. Males should be doing testicular exams now. Start now, once a month. Check, make sure you don't have any lumps. Mole checks. Look at the ABCDEs of your moles. Is it asymmetrical? What's the border look like? What color is it? What's the diameter? How big is it? And is it evolving? Practice abstinence or use condoms because HPV is linked to cervical and penile cancer. And so just taking care of yourself. And then there is a vaccine for that as well. So there are other things that you can do, but those are some of the big ones. So take care of yourself, be healthy, make good choices. And I think that's it. So if you have any questions about cancer, how it forms, what genes are involved, benign versus malignant, all that kind of stuff, just let me know.